Yeah, thank you, Angela and Bernardino, for letting us present some data today. Vesper is a clinical stage biotech company spun out of Aarhus University, where I'm one of the co-founders and chief medical officer. Our aim with this program is to develop an oral therapy for frontotemporal dementia by targeting sortilin. These are my disclosures, and just to be funny, I can say that I'm happy it can still stay on one page. All right, so we are targeting this disease, frontotemporal dementia, which is a fatal midlife, rapid developing form of dementia with a life expectancy of 7 to 10 years. Most of these cases are sporadic, but there is also genetic causes for frontotemporal dementia, where the three major causes or causal genes are granulin, MAPT, C9orf72. We are specifically targeting the granulin population. So some of the features that characterize FTD-GRN is decreased progranulin levels, lysosomal dysfunction, increased apoptosis, and increased neuroinflammation. So what does sortilin actually do? So sortilin is a 1TM receptor that was discovered in the mid-90s in our lab, and we have worked with it ever since, and it has given us gray hairs for many reasons. But one of the things that it does is that when you have FTD with granulin haploinsufficiency, you have reduced levels of progranulin, you have lysosomal dysfunction and neuroinflammation. Sortilin is the receptor that's shown right here, and we have developed over the last few years a small molecule that's able to inhibit that specific interaction between progranulins C-terminus and the neurotensin or NTIS1 binding site inside sortilin, the donut. And when we inhibit using this compound, we can increase the levels of progranulin. We can prevent gliosis and neuroinflammation based both on elevation of progranulin, but also directly by inhibiting sortilin, as it has a role in inflammation. Furthermore, not depicted on this schedule or this pictogram here, sortilin also have other functions or mode of actions. That is, that it can be part of a apoptosis signaling complex. This can also be inhibited by the compound. When we inhibit using the compound, we can also shuttle more or shunt more of the progranulin via another pathway to the lysosome, which is via LRP1 or mannose-6-phosphate receptor pathway, piggybacking on prosapacin. So a little more about the compound. So the compound, even though I'm not showing you the structure, it has some different attributes compared to, for example, an antibody approach. So if you look at the picture here on the left, we have measured in a cell model that if you use an antibody against sortilin, you will get downregulation. You remove some of the sortilin from the cell surface, also affecting other functions of sortilin, and also you will reduce the total amount of sortilin. We have used in this assay the highest possible amount of VES001, and we see absolutely nothing on the protein levels. And also, if we dose animals for 3 months up here, we do not see any change in the mRNA expression level of SORT1 in the compartments or the part of the brains that we looked into, both cortex, hippocampus, and thalamus. Furthermore, down the development path of this compound, we also did a rat microdialysis experiment. The reason for this was to figure out, does compound or enough compound get into the brain? So does it actually go to the right place? So in this experiment here, we have inserted 2 microdialysis probes where we can measure both the compound and also progranulin levels, and 2 catheters in the jugular vein and also the cisterna magna, where we can detect, as shown here above, and uncorrected, so not corrected for protein binding values up here. So we can see the compound levels in plasma, CSF, and hippocampus. And down here at the bottom, we have measured the progranulin levels in plasma, CSF, and in the hippocampus microdialysate. It's normalized to the baseline level. The reason for that is that the levels of progranulin inside, well, in plasma compared to CSF, is about 50 to 100 times higher in plasma. Furthermore, we tested the compound in a disease model of FTD-GRN. So this is the mouse model that's haploinsufficient in the granulin gene, so has lower exposure, or is basically. And what we see here is that with the compound, we're able to increase progranulin levels in plasma, both in wild-type animal, but also in the granulin haploinsufficient animal. And we have seen also from other studies and other places where similar things has been done, that these animals seems to be more sensitive to inhibition of sortilin to increase progranulin level. So, coming to the trial, we initiated then a clinical trial with VES-001 in healthy volunteers. This is a randomized double-blind placebo-controlled study with 78 healthy volunteers, where we have 48 in the single ascending dose study and 30 individuals in the multiple ascending dose study. We've used 6 dose groups in the single ascending dose groups and 3 dose levels in the last MAD study. We have also, as Angela, you referred to earlier, we need to know if the compound actually gets where it needs to go. So this is designed specifically to figure out how the compound behaves in humans, but also if it gets to the right compartments. So these healthy volunteers in the last 2 SAD cohorts and the entire MAD cohorts were subjected to 2 lumbar punctures at baseline and after the end of treatment. The endpoints for this study was safety and tolerability, and pharmacokinetics, and exploratory were pharmacodynamics. So most importantly, well, we're happy to see that there were no serious adverse events reported. We have few treatment emergent adverse events observed that were all graded mild in severity. We had no safety, chemistry, urine analysis, vital signs, or ECG events recorded. The top mild adverse events that we had observed was fatigue and headache. Going a bit into the pharmacokinetics of the compound, we saw that it showed a nice dose linear PK, both in the SAD and the MAD study. We have then a high resolution PK curve here and here. Not that you need to dissect it further, but the point was here to figure out whether we see any induction or inhibition of the compound, to see if there was any changes in the enzymes or proteins that handled VES-001. And there was none. So we see no induction or inhibition. These levels of compound in the middle here are the trough levels, so the lowest levels, and they are above in the 2 dose groups, above the target engagement ratio of about 55 nanomolar. In the figure to the right, we had lumbar punctures taken. I'm just showing the ones from the MAD study. Here we could see that the compound is able to get into CSF. Also here we have calculated a KPU of 1.6. That just basically means that the compound diffuses relatively unhindered into the brain compartment. So you calculate this by dividing the free fraction of compound in CSF, divide that with the free fraction of compound in plasma, then you get a KPU. If it's 0, nothing gets in. If it's 1, it equilibrates equally across, and 1.6 indicates that it could be transported actively. Furthermore, we found a half-life of around 11 hours, which gives us guidance to the dose regimen that we can go forward with. And lastly, we were able to detect progranulin increases, so we have target engagement in the plasma or the blood compartment, but also in the CSF compartment here. We know from previous data that it will take significantly more than 7 days to get to a steady state of progranulin when you do inhibit sortilin. It's more like 2 or 3 weeks. Oh, it's stuck. Okay, it jumps a bit. All right. If you take the value, so we have from the previous slide, we have around an increase of 0.4 nanograms per mil in CSF. And this is important for us, the CSF compartment, and less so the plasma compartment. But what we see here is if we take the data from the GENFI study and add this value of 0.5 nanograms per mil, we know that we will be in an area that's similar to a normative range for these individuals. But to get any closer to this, now it's stuck again.
No idea.
Yeah, try that. Now it moves, so don't. All right. We have just initiated, so just starting again from the previous slide. So to figure out what level of dose we need for VES001 to get the progranulin response we need to normalize the levels of progranulin in CSF and in plasma, we have initiated a phase 2 trial now in asymptomatic carriers of granulin mutations. It's an open label study. We will dose for 3 months, use 2 doses of compound. We will again take lumbar punctures 3 times during this study, so we have a baseline and the steady state levels of progranulin in CSF after both doses that we will subject the patients to. This study requires 6 carriers and is fully enrolled by now. We have 2 sites, Rotterdam University Medical Center in the Netherlands and University College London in the UK. And the first patient was dosed in January this year. And the primary endpoints is again safety and tolerability because we are moving over or dosing for a longer period of time. Again, VES001 pharmacokinetics, which I assume will be exactly the same as in a healthy volunteer, as it does not seem to be affected by granulin mutations. But granulin turnover might be changed in individuals with granulin mutations, where we need this study to fix in on the dose. And lastly, I would like to thank you for your attention in this dark room this late afternoon and with 0 coffee. But the summary is that we have shown that sortilin inhibition elevates progranulin. The mode of action is demonstrated in several species, also disease-relevant models. We have phase 1 clinical trial data from healthy volunteers that shows that the compound is safe. We have proof of concept, that means we have seen elevation of progranulin in plasma and CSF. And we have now started a phase 2 trial in granulin carriers, which is ongoing and will read out later this year for dose selection. And with that, I would like to thank you for your attention.